Hello everyone, welcome to the next lecture in our physiology video lecture series. This particular lecture is titled Respiration Part 2. In this lecture, we will look into the different aspects of regulation of the respiratory system as well as the various nervous centers present in our brain that are responsible for the same. So let us begin. So first we look at why regulation of respiration is essential. The nervous system normally adjusts the rate of alveolar ventilation almost exactly to the demands of the body so that the oxygen pressure or the oxygen partial pressure and the carbon dioxide partial pressure in the arterial blood are hardly altered even during heavy exercise and most of the types of respiratory stress. So consider the fact that whenever we are taking a rest or sitting in one position, the requirement of the muscles and different parts of our body towards oxygen and removal of carbon dioxide is met by our respiratory system. But during heavy exercise or in an active state, this is also the case that if in heavy exercise, when our muscles are working actively and different parts of our body are working more and more actively than normal, and thus the requirement of oxygen is higher in order to produce energy, then also the respiratory system is able to keep up with the increased need of oxygen and subsequent removal of carbon dioxide. How exactly is this particular change or the regulation of the respiratory system happens in order to match the respiratory rate with the requirement of the body is what we will look into in this particular lecture. The most important thing the most important part of our brain that is involved in regulation of respiration is the respiratory center. The respiratory center is composed of several groups of neurons located bilaterally, which means that if we have a line going right through the anterior posterior axis just like this, then on both sides of the anterior posterior axis, the respiratory neurons are present symmetrically in the medulla oblongata and the pons of the brain stem. So obviously we know that our brain is divided into three major parts: the cerebrum, the cerebellum, and the medulla oblongata and pons. The medulla oblongata and the pons is the part of the brain stem that connects the brain to the spinal cord. These parts of the brain are essentially involved in the involuntary actions of our brain, which is not directly under our control. That is similarly, that is in the case of respiration also. Regulation of respiration is not something that is directly in our control all the time. It is an involuntary event that occurs without our direct engagement into the effect, or without our direct involvement in the process. So the respiratory center is divided into three major collection of neurons. The first is a dorsal respiratory group located in the dorsal portion of the medulla, which mainly causes inspiration. So if we were to draw the three axes which determine which are present in our body, which is the anterior posterior axis, which will pass right through the brainstem, the dorsal ventral axis, which will pass perpendicular to the screen we are looking at, and the proximal lateral axis or the medial lateral axis, which will with the medial side closer to the center of the brainstem and the lateral side away. We will be able to see that particular neurons are present in the dorsal side or the upper side, or the side facing towards us on the screen at the moment, which are known as the dorsal, which is known as the dorsal side, and the neurons themselves are known as the dorsal respiratory group. The dorsal respiratory group is mainly responsible for the process of inspiration. Next, we look into the ventral respiratory group, located in the ventrolateral part of the medulla, which mainly causes expiration. So ventral means towards the opposite side of the screen. So if we imagine a three-dimensional structure, it will be facing opposite to us and we won't be able to see all of it. And lateral here means that it is located away from the center of the brainstem. And thus the ventral respiratory group located in the ventrolateral part of the medulla mainly causes expiration. Finally, the respiratory center consists of the pneumotaxic center located dorsally in the superior portion of the pons, which mainly controls rate and depth of breathing. So here we are shown all the different parts of the respiratory center, which include the dorsal respiratory group, which is present right here, the ventral respiratory group responsible for expiration, which is present right here, and finally the pneumotaxic center, which is present right in the superior part of the pons. So we move on and look at the dorsal respiratory group of neurons, which control inspiration and the respiratory rhythm. The dorsal group of respiratory neurons extends most of the length of the medulla. Most of its neurons are located within the nucleus of tractus solitarius. The nucleus of tractus solitarius is the sensory termination of both the vagal and the glossopharyngeal nerves, which transmit. So first we have to understand what before we go on, we have to first understand what nucleus means. In the central nervous system, a nucleus does not mean the nucleus of a cell, but instead a nucleus is the collection of the cell bodies of various neurons that are traveling along the CNS. So here the nucleus means the collection of cell bodies that are present in the tractus solitarius, which is in turn the sensory termination for both the vagal and the glossopharyngeal nerves, which transmit sensory signals into the respiratory system, originating from first the peripheral chemoreceptors and secondly the baroreceptors and several types of receptors present in the lungs. So in the previous lecture on regulation of blood pressure, we have already come across chemoreceptors and baroreceptors. Chemoreceptors are special receptors present in our body which detect the presence of or the increase or decrease in the concentration of oxygen, carbon dioxide, or pH of our blood. Similarly, baroreceptors are particular receptors present in our blood which detect the changes in blood pressure. Hence, these two types of receptors signal directly to the dorsal respiratory group of neurons in order to provide them the information about changes in the physiological conditions in our body. Next, we look into the rhythmical inspiratory discharges from the dorsal respiratory group. The basic rhythm of respiration is generally maintained in the dorsal respiratory group of neurons. Even when all the peripheral nerves entering the medulla have been sectioned and the brainstem transected both above and below the medulla, this group of neurons still emits repetitive bursts of inspiratory neuronal action potentials. Thus, these particular dorsal respiratory group of neurons will keep firing even though they have been isolated from the entirety of the brain. What this means is that the firing or the reactions of the dorsal respiratory group is independent of the signals coming from elsewhere in the brainstem or the body. The basic cause of these repetitive discharges is unknown. In primitive animals, neural networks have been found in which activity of one set of neurons excites a second set, which in turn inhibits a first set. Then after a period of time, the mechanism repeats itself, continuing throughout the life of the animal. So this kind of a close feedback loop will be able to produce the inspiratory rhythmic discharges that might be necessary for the continuing of respiration. Thus, this has been shown in primitive animals, but these repetitive discharges have not been shown in humans. Therefore, most respiratory physiologists believe that some similar network of neurons is present in human beings located entirely in the medulla, which is responsible for the basic rhythm of respiration. Then they believe that it is located entirely within the medulla because obviously if we isolate the medulla from the brainstem, the respiratory discharges still continue, and thus the whole system or the whole closed feedback loop must be present within the medulla itself. Next, we move on to the inspiratory ramp signal. So we have to understand that our breathing continues, our lungs fill at a continuous rate till they are completely full. That is the process of inspiration, after which they let go of the air present in them, and that is expired out of the lungs. This does not happen in a haphazard or a sharp manner, but the lungs filled continuously for some time and then discharge or expire air continuously for some time. Thus, the process of respiration occurs not in bursts or gasps, but at a continuous rate. This is important because we will next look into why this occurs or how is it useful. The nervous signal that is transmitted to the inspiratory muscles, mainly to the diaphragm, is not an instantaneous burst of action potentials. Instead, in normal respiration, it begins weakly and increases steadily in a ramp manner for about 2 seconds. Then it ceases abruptly for approximately next 3 seconds, which turns off the excitation of diaphragm and allows the elastic recoil of the lungs and the chest wall to cause expiration. Thus, inspiratory signal is a ramp signal. The obvious advantage of the ramp is that it causes a steady increase in volume of the lungs during inspiration rather than inspiratory gasps. So, first we have to understand what a ramp signal exactly means. We have already seen that what action potential is. It is a sharp peak in it occurs as a sharp peak in change, sharp peak which denotes the change in membrane potential of a nerve, and which then transmit to the membrane sharp peak in change, sharp peak which denotes the membrane potential change in the muscle fiber. So it will look like a sharp peak like this, but this is not what the inspiratory signal looks like. It is not one isolated action potential, but a continuous number of action potential that occurred one after the other. So you can imagine it as a one action potential followed by 2, followed by 3, followed by 5, so that the rate of action potential that occur or are transmitted to the diaphragm increases continuously, and thus we have an inspiratory ramp signal which ramps up little by little till the diaphragm needs to be completely extended in order for the lungs to inspire properly. This was the inspiratory ramp signal. Next, we'll look into the role of pneumotaxic center in the inspiratory ramp signal. The pneumotaxic center limits the duration of inspiration and increases the respiratory rate. The primary effect of this center is to control the switch off point of the inspiratory ramp. So how long will this continuous increase in action potentials occur, or how long will the ramp signal occur? Thus, controlling the duration of the filling phase of the lung cycle. When the pneumotaxic signal is strong, inspiration might last for as little as 0.5 seconds, thus filling the lungs only slightly. When the pneumotaxic signal is weak, inspiration might occur for 5 seconds or more, thus filling the lungs with a great excess of air. Thus, the reactions of the pneumotaxic center is inversely proportional to the time period of inspiration. The function of the pneumotaxic center is primarily to limit inspiration. This has, however, a secondary effect, which is the increasing of rate of breathing. Because limitation of inspiration also shortens expiration and the entire period of respiration. Thus, as the time period of inspiration is getting decreased with increasing pneumotaxic center signals, this will cause the overall cycle of respiration to be shorter. Thus, if the overall inspiration and expiration cycle of respiration is shorter, then more number of these cycles can occur within a minute, and hence our breathing rate increases. A strong pneumotaxic signal can increase the rate of breathing to 30 to 40 breaths per minute, whereas a weak pneumotaxic signal may reduce the rate to only 3 to 5 breaths per minute. Next, we look into the ventral respiratory group of neurons, which function in both inspiration and expiration. The ventral respiratory group of neurons, as we have already discussed before, is located in each side of the medulla, about 5 millimeters anterior and lateral to the dorsal respiratory group of neurons. So, what anterior means a bit along the anterior posterior axis, and lateral means away from the center of the brainstem. The function of this neuronal group differs from that of the dorsal respiratory group in several important ways. These ways include the neurons of the ventral respiratory group remain almost totally inactive during normal quiet respiration. There is no evidence of ventral respiratory neurons participating in the basic rhythmical oscillation that occurs during respiration. When the respiratory drive for increased pulmonary ventilation becomes greater than normal, that is when the body is moving from a resting phase to an active phase, the respiratory signals spill over into the ventral respiratory neurons from the basic oscillating mechanism of the dorsal respiratory area. Electrical stimulation of a few of the neurons in the ventral group causes inspiration, whereas stimulation of others causes expiration. Thus, we can imagine the ventral group of respiratory neurons as a backup mechanism, which, when there is a need for excess rate or depth of breathing, comes into play when the dorsal group of neurons fires so rapidly that the respiratory signals spill over into the ventral side and thus activating this ventral respiratory group of neurons. Now we will change gear, and after looking at the normal levels of control or regulation on the respiratory system, we will see how external factors are detected by the respiratory center and the respiratory rate changed accordingly. So we will look at the regulation of the overall respiratory center activity. First, we look at the chemical control of respiration. The ultimate goal of respiration is to maintain proper concentration of oxygen, carbon dioxide, and hydrogen ions in the tissues. And thus, obviously, the respiratory activity is highly responsive to changes in each of these. Excess carbon dioxide or excess hydrogen ions in the blood mainly act directly on the respiratory center itself, causing greatly increased strength of both inspiratory and expiratory motor signals to the respiratory muscles. Thus, an increase in carbon dioxide or hydrogen ions affect the respiratory center directly, which increases the rate of expiration and inspiration. Thus, the overall rate of respiration or breathing is increased. The reason behind this is that the respiration, increase in carbon dioxide and hydrogen ion levels mean that there is an accumulation of carbon dioxide and hydrogen ions in the blood. This signals to the brain that the expiration rate needs to be increased so that more of the carbon dioxide will be blown out into the atmosphere out of our body. And thus, the primary reaction towards increase in carbon dioxide and hydrogen ion concentration is to increase the rate of breathing. Oxygen, in contrast, does not have a significant direct effect on the respiratory center of the brain in controlling respiration. Instead, it acts almost entirely on the peripheral chemoreceptors located in the carotid and aortic bodies. The carotid and aortic bodies are small organoids that we have already come across before. We have looked into them during our regulation of blood pressure lecture and found out that these particular organoids signal directly via the hearing nerve to the brain in order to control blood pressure. They come up here again in regulation of respiratory signal as well. And these in turn, that is the carotid and the aortic bodies, transmit appropriate nervous signals to the respiratory center for control of respiration. Thus, we see there is a stark difference between the regulation of respiration in response to changes in carbon dioxide and in response to changes in oxygen. In case of carbon dioxide, the respiratory center is directly involved and the changes are detected directly at the respiratory center itself. Whereas in case of oxygen, the changes are detected at the level of peripheral chemoreceptors, which then signal to the respiratory center. Thus, the sensing of the changes in oxygen concentration is indirect in this case. So we will look into the direct chemical control of respiratory center activity by carbon dioxide and hydrogen. It is believed that none of the previously mentioned respiratory center, that is the dorsal, ventral, or the pneumotaxic center, are affected directly by the changes in blood carbon dioxide concentration or hydrogen ion concentration. Instead, an additional neuronal area, which is known as the chemosensitive area, is located bilaterally, lying only 0.2 mm beneath the ventral surface of the medulla. This area is highly sensitive to changes in either blood CO2 or partial pressure of blood CO2 or hydrogen ion concentration, and it is in turn it excites other portion of the respiratory center. So now we'll look into in detail the excitation of the chemosensitive neurons by hydrogen ions, which acts as the primary stimulus in its signaling. The sensor neurons in the chemosensitive area are essentially excited by hydrogen ions. In fact, it is believed that hydrogen ions may be the only important direct stimulus for these neurons. So these neurons detect changes in hydrogen ion concentration only. However, hydrogen ions do not easily cross the blood-brain barrier. So obviously we know that between the normal circulation of blood and the brain, there is a particular barrier known as the blood-brain barrier or BBB present, which does not allow normal contaminants present in the blood to come into contact with the brain. This blood-brain barrier also hinders the transport of hydrogen ions directly into the brain. And thus presents a major problem, because as we have already seen or read that the chemosensitive area is responsive only to the stimulation of hydrogen ions, but hydrogen ions themselves can travel to the chemosensitive area. For this reason, changes in hydrogen ion concentration in the blood have considerably less effect in stimulating the chemosensitive neurons than do changes in blood carbon dioxide. Even though carbon dioxide is believed to stimulate these neurons secondarily by changing the hydrogen ion concentration. So how does carbon dioxide affect the chemosensitive neurons by indirectly changing the hydrogen ion concentration is what we will look into in the next slide. So now we'll look into the stimulation of the chemosensitive area by hydrogen ions formed by carbon dioxide. So this is a schematic showing the whole entire process. Although carbon dioxide has little direct effect in stimulating the neurons in the chemosensitive area, it does have a potent indirect effect. It does this, that is the stimulation of the chemosensitive area, by reacting with water or the tissues in order to form carbonic acid, which dissociates into hydrogen and bicarbonate ions, which is the reaction which is shown right here. Carbon dioxide plus water forms carbonic acid, which then breaks into hydrogen ions and bicarbonate ions. These hydrogen ions, which are now produced, have a potent direct stimulatory effect on respiration. The reactions are shown in this figure. But the question remains, why does the blood carbon dioxide have a more potent effect in stimulating chemosensitive neurons than do blood hydrogen ions? The answer is that the blood-brain barrier is obviously not very permeable to the hydrogen ions. But carbon dioxide passes through this barrier almost as if the barrier did not exist. Consequently, you can imagine a scenario in which the blood-brain barrier is present somewhere in the middle of this particular carbonic acid forming step, or rather here, before even the carbonic acid is formed. The carbon dioxide first passes right through the blood-brain barrier and then reacts with water in order to form carbonic acid, which then dissociates it to give hydrogen ions and then bicarbonate. Thus, essentially, we have been able to transport hydrogen ions from one end of the blood-brain barrier to the other. And these hydrogen ions, which are produced via the increased carbon dioxide levels, is what affects the chemosensitive area. Consequently, whenever the blood PCO2 or partial pressure of CO2 increases, so does the partial pressures of CO2 of both the interstitial fluid of the medulla and the cerebrospinal fluid. In both these fluids, the carbon dioxide immediately reacts with water in order to form new hydrogen ions. Thus, paradoxically, more hydrogen ions are released into the respiratory chemosensitive sensory area of the medulla when the blood carbon dioxide concentration increases than when blood hydrogen ion concentration increases. Thus, the entire process is necessarily dependent on the changes in blood carbon dioxide level than hydrogen ion, even though the chemosensitive area is much more sensitive or reacts much better towards stimulation by changes in hydrogen ion. Next, we look into the effects of PCO2 and the arterial pH on rate of alveolar ventilation. So the partial pressure of CO2, or later we can say the concentration of carbon dioxide present in the bloodstream, is shown in red, and the changes in pH or concentration of hydrogen ions is shown in green. This figure shows the quantitatively the approximate effects of blood partial pressure of carbon dioxide and blood pH. And we know that pH is essentially the inverse logarithmic measure of hydrogen ions concentration and can act as a proxy for hydrogen ion concentration directly. Note especially that the very marked increase in the ventilation caused by increasing PCO2 beyond the normal level. So normal level is given here in black, And just as we cross the normal level of PCO2, we see a sharp increase in the blood in the alveolar ventilation, and thus we see a sharp increase in ventilation caused by increase in PCO2 in normal range between 35 millimeters of mercury to 75 millimeters of mercury. This demonstrates the tremendous effect of carbon dioxide changes having controlling the respiration. On the other hand, the changes in respiration in the blood pH range of 7.3 to 7.5 is less than 1/10 as great. So pH changes do not affect the rate of alveolar ventilation as much as changes in carbon dioxide do. And thus we can confirm blood carbon dioxide concentration is the major plays the major role in controlling the alveolar ventilation via stimulation of the chemosensitive neurons. Next, we look into another point, which is the unimportance of oxygen for control of the respiratory center. Changes. So we have already seen how carbon dioxide and hydrogen affect directly the respiratory center. But now we'll see that oxygen does not actually play an important role at all in affecting the respiratory center directly. Changes in oxygen concentration have virtually no direct effect on the respiratory center itself to alter the respiratory drive. The reason behind this is that the hemoglobin oxygen buffer system delivers almost exactly the normal amounts of oxygen to the tissues, even when pulmonary partial pressure of oxygen changes from a value as low as 60 millimeters of mercury up to a value as high as 1000 millimeters of mercury. Therefore, except under special conditions, adequate delivery of oxygen can occur despite changes in lung ventilation ranging from slightly below 1/2 normal to as high as 20 or more times normal. Thus, if the regulation was based on oxygen, the system would not be able to detect any considerable problems with the lung ventilation itself, because the hemoglobin oxygen buffer system is so efficient that it is able to deliver normal amounts of oxygen even if the oxygen concentration drops to half its normal value, or the rate of ventilation increases to 20 times its normal value. And thus the efficiency of the hemoglobin oxygen system renders it such that the oxygen concentration cannot be used as a very good measure, as a measure at all, for maintaining respiratory rate, at least by the direct stimulation of respiratory center. This is not true for carbon dioxide, because both blood and tissue partial pressure of carbon dioxide changes inversely with rate of pulmonary ventilation. That is, the concentration of carbon dioxide is not so actively maintained, but the higher the rate of alveolar ventilation, the more carbon dioxide is pushed off or expired out of the lungs. And thus it gives us an almost linear relationship, that is the partial pressure of carbon dioxide present in the blood gives us an almost linear relationship with the rate of rate of respiration that is occurring. And thus the processes of animal evolution have made carbon dioxide the major controller of respiration and not oxygen. Next, we will look into the role of the peripheral chemoreceptor system for control of respiratory activity and role of oxygen in respiratory control. So here we see a schematic diagram showing us the respiratory center and the carotid and aortic bodies that are present in various parts of our circulatory system, and how they are connected via the glossopharyngeal nerve and the vagus. Special nervous chemical receptors called chemoreceptors are located in several areas outside of our brain. Most of the chemoreceptors are in the carotid bodies, however, a few are also in the aortic bodies. So we have already looked through the carotid bodies and the aortic bodies in a previous lecture on blood pressure regulation, and we have seen how these particular chemoreceptors sense and detect the changes in levels of pH, the partial pressure of oxygen, and the partial pressure of carbon dioxide that are occurring in our body. Each of the chemoreceptor bodies receives its own special blood supply through a minute artery directly from the adjacent arterial trunk. Further, blood flow through these bodies is extreme, 20 times the weight of the bodies themselves each minute. Therefore, the percentage of oxygen removed from the flowing blood is virtually zero. This means that the chemoreceptors are exposed at all times to arterial blood and not venous blood, and their PO2s are the same as arterial PO2s. What this means is that the carotid bodies are very small and the blood flow rate through them is extreme. What this causes is that the carotid bodies and the blood vessels and the tissues surrounding them are not able to take up oxygen at the extreme rate at which blood is flowing through them. Thus, when blood flows through the carotid and the aortic bodies directly, the blood that is flowing through them is the arterial blood. That is, this blood has not undergone any capillary exchange, and the nutrients present in the blood is the same as it was when it was pumped out of the heart. And thus, when the carotid bodies or the aortic bodies detect the presence or the concentration of oxygen present in the blood, they are detecting the arterial concentration of the blood and not the venous concentration of the blood that would have occurred, that would have been measured if the blood flow wouldn't have been this extreme, causing the blood to undergo blood nutrients to undergo exchange with nearby tissues, and thus lowering the actual concentration of oxygen that is actually being pumped by the heart. Next, we look into the stimulation of the chemoreceptors by decreased arterial oxygen. When the oxygen concentration in the arterial blood flow falls below normal, the chemoreceptors become strongly stimulated. This is demonstrated in the figure that is shown right here, which shows us the effect of different levels of arterial PO2, which is the variable that is plotted on the x-axis, on the rate of nerve impulse transmission from a carotid bodies, which is plotted on the y-axis. Note that the impulse rate is particularly sensitive to changes in arterial PO2 in the range of 60 down to 30 millimeters of mercury, which is shown by the steeper portion of the graph that ends about here. A range in which hemoglobin saturation with oxygen decreases rapidly. So we have already seen that even though the rate, blood pressure might, the arterial partial pressure of oxygen might reduce to half its normal value or increase to 20 times excess of its normal value, the hemoglobin oxygen buffer system is efficient enough to still deliver in required amounts of oxygen to different parts of the body. But when the partial pressure of oxygen further decreases, that is moves from down from 60 millimeters of mercury to about 30 millimeters of mercury, the efficiency of the hemoglobin buffer system decreases rapidly, and hemoglobin saturation with oxygen also decreases rapidly. This is when the chemoreceptors start detecting and firing at a rapid rate, which can be shown right here, where the normal rate of firing can be around 100% at 100 millimeters of partial pressure of oxygen, but which increases little by little up to around 700%, the normal rate of firing, where the partial pressure of oxygen keeps on decreasing under the 60 millimeters of mercury mark. The extract mechanism by which hypoxia, which is the event in which oxygen concentration decreases in our blood, is detected by the carotid body chemoreceptor neurons is not known. But there have been speculations regarding this particular theory, regarding this particular event, in which how the carotid bodies sense the decrease in oxygen concentration. One of the theories that are proposed of the decrease in oxygen concentration, that of the detection of decrease in oxygen concentration, is that the neurons and different cells in the which make up the carotid bodies have an extremely high rate of metabolism, and extreme, and thus process a lot of mitochondria as well, which process oxygen at a high rate. When the oxygen concentration decreases below it decreases below a particular level, the rate of metabolism of these particular carotid body cells decreases rapidly. This decrease in the metabolism of the carotid body cells is what signals to the chemoreceptor neurons via the hearing, via the vagus and the glossopharyngeal. This is one of the theories that was proposed to explain the mechanism by which hypoxia is detected by the carotid body chemoreceptor neurons. But there is not enough conclusive proof, and this is still being researched into. An increase in either carbon dioxide concentration or hydrogen ion concentration also excites the chemoreceptors, and in a way indirectly increases the respiratory activity. Thus, the chemoreceptors can also detect the changes in carbon dioxide and hydrogen ion concentration. However, the direct effects of both these factors in the respiratory center itself are so much more powerful than their effects mediated through the chemoreceptors, which, for comparison, in the carotid bodies, the effect is around 7 times lower than the effect that is caused directly at the respiratory center. That for practical purposes, the indirect effects of carbon dioxide and hydrogen ions through the chemoreceptors do not need to be considered. And thus, for practical reasons, we can consider that the carotid bodies and the chemoreceptors are directly stimulated by only decreasing arterial oxygen partial pressure, and is not very much affected by the changes in carbon dioxide and hydrogen ion concentration. Next, we look into the effect of low arterial partial pressure of oxygen to stimulate alveolar ventilation when arterial carbon dioxide and hydrogen ion concentrations remain normal. The figure that is shown here shows the effect of low arterial partial pressure of oxygen on alveolar ventilation when the partial pressure of carbon dioxide and hydrogen ion concentrations are kept constant at the normal levels. So in this particular plot, we are keeping the partial pressure of carbon dioxide constant and only changing the partial pressure of oxygen. In other words, in this figure, only the ventilatory drive due to effect of low oxygen on chemoreceptors is active. So we can see only the effect of oxygen concentration on the chemoreceptors themselves. The figure shows almost no effect on ventilation as long as arterial PO2 remains greater than 100 mm of mercury. The reason behind this is something we have already discussed, because the hemoglobin oxygen buffer system acts so efficiently at PO2 above 100 millimeters and much more above than that the body is not very much concerned with changes above 100 millimeters of mercury, and thus the rate of ventilation is not affected as long as the actual PO2 remains greater than 100 millimeters of mercury. But at pressures lower than 100 millimeters of mercury, ventilation approximately doubles when the arterial PO2 falls to 60 millimeters of mercury. So we can see from a alveolar ventilation normal rate at 1, when we reach at 60 millimeters of mercury, 60 millimeters of mercury of partial pressure of oxygen, the rate is almost doubled at 2, and can increase as much as 5-fold at very low PO2. So as the partial pressure of oxygen keeps on decreasing, we see a very sharp rise in the alveolar ventilation. Under these conditions, low arterial PO2 obviously drives the ventilatory process quite strongly. And thus, in this using this particular plot, we are able to isolate how only the partial pressure changes in oxygen levels affects the ventilation when we keep the PCO2, or the partial pressure of carbon dioxide and hydrogen ion concentrations, relatively constant. Finally, we look into something known as acclimatization, which is chronic breathing of low oxygen stimulates respiration even more, which is a phenomenon known as acclimatization. Mountain climbers have found that when they ascend a mountain slowly over a period of days rather than a period of hours, they breathe much more deeply and therefore can withstand far lower atmospheric oxygen concentrations when they ascend rapidly. This is called acclimatization. The reason for acclimatization is that within 2 to 3 days, the respiratory center in the brain stem loses about 4/5 of its sensitivity to changes in PCO2 and hydrogen ions. Therefore, the excess ventilatory blow-off of carbon dioxide that normally would inhibit an increase in respiration fails to occur, and low oxygen can drive the respiratory system to a much higher level of alveolar ventilation than under acute conditions. So what does the whole process mean? So what happens is that in normal conditions, the rate or the concentration of carbon dioxide and hydrogen, which was, as we have seen, 7 times more powerful than that of the concentration of oxygen that is detected by the chemoreceptors, is what drives the respiratory rhythm. But as a person ascends a mountain slowly, within 2 3 days, the brain is able to adapt to the mountain conditions and is able to understand that the concentrations of the concentration changes in the CO2 and hydrogen ions is not able to effectively determine or regulate the respiratory rate. This is because in the low oxygen conditions, the carbon dioxide concentration and the hydrogen ion concentration still mask the respiratory regulation, and the respiratory rhythm thus cannot increase, as increasing the respiratory rate will cause more blow off of carbon dioxide, which signals to the respiratory center directly and thus prevents an increasing respiratory rate. But as this continues for some time, the brain is able to lose its sensitivity little by little towards the carbon dioxide and hydrogen ion concentration levels. And in this condition, a person is known as acclimatized. When the brain now starts determining the level of respiratory rate depending on the concentration of oxygen alone. That is the masking effect of the carbon dioxide and hydrogen ion levels are not taken into consideration anymore, and only the low oxygen levels can drive the respiratory rate, and thus there is an increase in respiratory rate even at the cost of decreasing the carbon dioxide levels in the blood. And this increase in respiratory rate is what allows a person to withstand far lower atmospheric oxygen concentration as they ascend higher and higher up a mountain. This whole process or this whole phenomenon is known as the process of acclimatization. We will look into details in the process of acclimatization in the next lecture, which will be on high altitude physiology, which will look into high altitude physiology as a part of it, and we'll go through that when we'll go through acclimatization in more detail when we go through it. So this was all we had for today. The references for these particular slides are the chapter 41 of the textbook of medical physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to this video.